Okay, so good afternoon. Thanks for coming. I guess there were a lot of interesting alternatives. And so what I'm going to do today is give you a little bit of a glimpse into neuroscience of memory, and I will combine very general stuff with some of the stuff we do in our lab. And so this can be fairly interactive. So if you have questions or you have idea or you have things you're interested in, please stop me. And in fact, so this can also go in a very different direction than I planned. So I have hundreds of slides on my computer so we can go in a different way. So now, first thing we have to say about memory is that it's not just one thing. And so this is the, very textbook image, but that gives you an idea of how neuroscientists and psychologists think about memory. And the first distinction you make is between declarative and non-declarative memories. So declarative means available to consciousness or something you can actually verbalize, so you can talk about. Whereas a motor skill, so for example, if you learn to do a back shot at tennis, that is also a memory. However, it's not something you can precisely explain in words, right? Otherwise, we would all be Roger Federer, right? So Roger would explain us how to do the back shot, and we will all do it perfectly. That's not the case. So this is important. This comes from psychologists, and it's important for us neuroscientists because different brain structures and brain processes are involved in these two things. So within declarative memories, so we make another very important distinction, and that is between memory about your personal history, so what we call autobiographical or episodic memory, and memory about facts, right? So for example, you may know that Paris is the capital of France, but you may or may not remember the day in maybe primary school when you learned that fact, okay? But the important thing, so one important thing and one thing we'll go into is that one type of memory, so this memory about facts, is always derived from experience, right? So you always learn a fact within an episode in your life. And then gradually you lose track of the episode, but you remember the fact, okay? Make another example. So you all know that on September 11th, 2001, there was a major attack on the United States. And most likely, many of you remember living through that day, right? So what's the difference? It's the emotional value, right, of the memory. So emotion is a very powerful drive that actually steers what will be remembered and what will not be remembered. Okay? So, and if you are a neuroscientist, what you are interested in is how that happens. So what are the mechanisms, what are the biological basis for that? Now, so into going in about what is memory. So I'm going to start from a little bit afar and so on, a very strange kind of memory. And this actually very important Dutch guy, so Nico Tinbergen, so was one of the founding fathers of ethology, right? The study of animal behavior. And so one thing he did, he looked at animals in a prairie and he tried to fly over them different sort of silhouettes. And he could find that animals would react by escaping to some of them, but not others. And the interesting thing is that even very young juvenile animals would do that, that probably never had an experience with birds of prey. So this is a sort of phylogenetic memory, as Tinbergen named it, that has to do with something, some pattern of memory that might be somewhere in our genes. So that was the claim. So that means something that you don't have to learn. Now, everything else, you actually have to learn. Now, and what you learn has actually a profound effect also in what you perceive and how you see the world. So let's make an interesting, so let's show an interesting example. So this is a memory task where I'm going to ask people to remember position on the chessboard, right? And positions on the chessboard can be on this kind, or they can be of this kind. Now, who are the chess players among you? So what's the difference between these two? Huh? Yeah. So actually, this one is not even a legal position, right? So you can have two white bishops on white cases, for example. Okay, now here's the interesting twist. So the researchers had this kind of task, and they tested normal people that actually are naive at playing chess, like me, and chess masters. Now look, this is what happens. If you test people on legal positions, then the masters are vastly better than naive people. If you test them on random positions, the masters do worse. Right? So why is that? Well, this is a very interesting idea that comes from psychology, and we neuroscientists are trying to understand. That is the idea of schemas. So a schema is a way the brain has to organize knowledge so that new information can be interpreted in the light of that knowledge, right? So if you are, so those of you who studied biology, for example, they had a much easier time at remembering what I say when I start talking about neurons, synapses, and neurotransmitters, and hippocampus and stuff like that, than if you studied theoretical physics or classic languages or so, and the other way around, right? So if you try, when a master is trying to encode a random position, the schema is getting in the way. So they will actually try to remember something that fits the schema. And so the errors they will make will go likely in the direction of making that position more sensible, right? More legal. So how does it work with the schema? So the idea is that we have some sort of division of labor in the brain. So our current view of memory systems as far as declarative memory goes, is that there are actually two memory systems. There is one memory system for storing information in the immediate. And there is another memory system to actually remember facts for the long term. So the first system would actually be in this structure here called the hippocampus, which is something that is buried pretty much behind your nose and is as big as your thumb. It's a relatively small structure in humans that looks a little bit like a seahorse if you section it in the right way, hence the name. Now, the hippocampus is what will make memories, right? So it's where memories first go. If you don't have an hippocampus, or if your hippocampus has been lesioned or is non-functioning, you might very well remember what happened in your early life, for example, but you would be completely incapable of forming new memories, right? So there is a story of a very famous patient, this guy Henry Molaison, or as it was known as HM. And this guy was operated for epilepsy in the 50s, and the surgeons at that time were fairly generous when it came to cutting. So the surgeon took out the entire hippocampus, both sides, the amygdala, etc. And so this guy was pretty much cured from epilepsy, but he got this very interesting death. So he could tell everything that happened in his childhood, okay? You could talk to him, visit him. He was completely okay, right? So if you were to talk to him for 1 hour, then just go to the restroom, you could come back, and he would just go, oh, hi, nice to meet you, Henry Molaison. I had no idea that he met you before, okay? So now, that's where memories are formed, the hippocampus. So the rest, so the cortex, which is way larger in humans, is where a lot of the facts are stored. And the idea is that there is an interaction between these two systems that happens by and large offline. And so the idea is that this sleep replay that we talked about is what would fuel it, right? Now, want to study the brain and want to study memory, as I told you, much of what we do has to be done in animals because we need to use invasive techniques. There are some exceptional cases where we can do a little bit of that with humans, but it's very little. So if you want to study declarative memory in animals, so memory is about things you can talk about, you have a fairly small problem that animals cannot talk. So they cannot tell you what they had for breakfast this morning. So how do you study? Well, you have to work by analogy. Now, a first analogy is about spatial memory. That is, memory about the location of things. So why is that why is that thought to have anything to do with memory about our life history? Well, the idea is that when we form a memory about an episode in our life, it's most likely to be spatially organized. So Tolman, who was a psychologist in the 40s, talked about a cognitive map, which meant that if you try to remember what happened today, you probably remember this room, you remember I was here, you remember there were some orange doors to the left, you remember who was sitting next to you, and everything is spatially arranged. Okay? So how can you study spatial memory in animals? So this is the most classic task. So this is a swimming pool. It's a small swimming pool for rats or mice. And there is water, and the water is made opaque. You can put in there some white paint or, for example, powdered milk until you can't see anything. And actually, there is a submerged platform somewhere in the swimming pool, which is the only place where animals can touch, and so they can actually find a safe haven. And so you can train animals to find this platform, and so they have to remember the position. So what's the nice thing about this? The nice thing is that rats and mice are extremely olfactory animals, which means that if you were to do it on a dry surface, they could probably just follow some odor scent. What probably animals would do is once they find the location that they're interested in, they would just pee on that and then just follow the urine scent. So in water, of course, they cannot do that. So what they have to do is find the position of the platform based on the position of landmarks in the environment. Right? For example, here there is this chessboard, here is the triangle, and the platform is a little bit here in the middle. So this is called the Morris water maze from Richard Morris, so the British neuroscientist who invented it. And just as for autobiographical memory, the hippocampus is actually involved. So if you lesion the hippocampus, the animal won't learn it anymore. So this is how it looks like. So the first time you do it with a normal animal, so the first time they would just circle, search around until they find the platform. After 10 trials, you leave it there, boom, they just go here. Rat with hippocampal lesion, after many trials, still circling it around. Now, another way to do it, which is one thing we actually do in the lab, is to use the fact that lab animals are eminently bored. So they actually like new things a lot because their existence is not that eventful. So it's not bad. And that means they like novelty. Now what you can do is, for example, you put them in a box with two objects, and you let them explore. They will explore it for a little bit, and then they will get bored by that as well. The next day, you put them in the box with in the same box with the two objects, but now one object has been moved. Now the idea goes, which works very well, and is something that has been done for years, is that if they remember the training, you can actually, they will explore more this object. Why? Because it's more novel. Right? So it's just more interesting because it's something they haven't experienced before. And that's what they do. So this is what it looks like. So these are rats and mice, which look a little bit alike. They are very different creatures, as you can see from the patterns. Rats are very laid back, etc. And mice tend to be a little hyper. I mean, also because this guy is more likely to be a prey than this guy. Actually, rats eat mice, which is a problem when, so in the lab, we have to keep them very well separated also because, so just the smell of rats will spook mice. So, so one thing we do is we use this other training procedure. And so what happens here, look, so during training, so the first day of the experiment, so we put the animals in the box 5 times for 5 minutes each time, but with different configurations of objects. So the first time, you have objects here and here, then it's here and here, here and here, and so on and so forth. So what you notice is that one position was always there, and the other one changes. Then the testing is actually the next day, the same configuration as the last one in training is used. So that means that if they only remember the last one, there shouldn't be any difference, right? Because these are identical. However, if they remember the entire series, then this one should still be more novel than this one because it was only present at training 1/3 of the times, and this one was present 100% of the times. So that means we are teaching animals to abstract, in a sense, over multiple episodes and to learn a probability distribution. And we think that this is one component into learning a schema. Okay? It's not the whole story, which is extremely tricky to do in animals, of course. So I cannot teach rats to play chess. But it's one step in that direction. Okay? So that I look at how animals can learn probability distributions, which turns out they can do pretty well. So a rat will learn this memory with 5 trials. And mice that are more stupid, they need about 20 over 4 days. So now let's go now to one from the behavioral point of view to something that is much, a little bit more detailed, and that is what neurons in the hippocampus do. Now, as I showed a little bit in the first presentation, so the hippocampus has these place cells. So these are neurons that will activate whenever the animal is in one specific place, for example, in this room, right? And that we symbolize, we represent with this what we call firing rate maps. So each one of these circles is the representation of a round arena or a round box. Typically, these things are like 1 meter across. So you have animals going, running around in this arena. You sprinkle a little bit of food on the floor, and they will go like Pac-Man, try to vacuum all the food. And this is what we call the firing map for a neuron. And so you see that this neuron likes sort of the northeast side of the arena, and this one likes the south side, and this one likes also the south side, this likes the north side, and so on and so forth. So if you record enough neurons, you have complete coverage of the arena. So the way this works, so this is another movie, so this comes from Bruce McNaughton's lab. I hope the sound works. So you'll see, so this is a rat, and this is a circular arena. So they have a little implant on their head with a wire going to the ceiling. So also the whole thing is seen from above. So this was done in semi-darkness, so rats are nocturnal animals. So you typically don't like to be in full-on light. So that's why the video itself is a bit dark. And then you will hear a click each time one neuron recorded fires. And see that. And again, so you see how precise this neuron is. So it never fires outside of this area. Right? So this is essentially how in 1971 John O'Keefe discovered these place cells. So he had an electrode in the rat brain and the hippocampus. So that was connected to an amplifier. The amplifier was connected to a loudspeaker. So he was actually listening to that live and was trying to figure out what would make these neuron fires, and then it had to conclude it was place. So what do you do then? Well, you have to do a lot of controls. You try move the furniture around to say it's not this particular piece of furniture that does it. You try to control for odors. You spray everything with vinegar and alcohol to, seriously, not to erase all odors, and so on and so forth. You have to do a lot of controls. But in the end, he concluded it can only be space. Now, this is one type of place code in the brain, so it's kind of the most obvious. And then, 2005, another group in Norway by Edvard and May-Britt Moser found this other type of firing rate maps in a brain structure that is a little bit close to the hippocampus. It's called the entorhinal cortex. And you see what this does is that now these neurons are firing in multiple places. And the striking thing is that these places are organized on a triangular grid. So this is, and so these are the so-called grid cells, which we'll see in a moment are actually extremely important. So John, Edward, and May-Britt got, for this thing, got a Nobel Prize. So it's considered one of the main success stories we have in neuroscience. Another type of fascinating cells is what's called head direction cells. And this is, if this is the GPS, so this is like the compass. So these are cells that will fire regardless of where you are, but depending on where your head is pointing to. So where it's pointing north, south, east, or west, different sets of cells will activate. So all together, so these are components of our, yeah.
But also social place cells. You've heard about that?
Yeah. Yes.
Also velocity cells.
There are a number of others, yes. So a social place cell is the social place cell is a cell that is firing not according to where you are, but according to where your mate is. So that they look at, and actually interestingly, those experiments were done in flying bats by the group of Nahum Ulanovsky, and which is a very interesting experiment because you can actually now see these place cells in 3D. Right? So see that now you have a cell that actually fires here, another one that fires here, and so on and so on. So this is the idea of the cognitive map, right? So this has been there since the 40s. So then O'Keefe, with the American psychologist Lynn Nadell came up with the idea. So they wrote a book in 1978 saying, oh, okay, now we know where this cognitive map is. It's in the hippocampus. Okay? So, and what's the advantage of having a map is that you can actually do geometry on it, right? So if I learn the layout of this room and I have a map-like representation, I can do geometry, which means I can do detours. So I know that, for example, if this path this way is obstructed, I can go this way, right? If all I did was learning how to get from here to here, so that I have to do 10 steps in this direction. So if there was an obstacle here, then my memory would be completely useless, because I would go 10 steps, after 5 I would be blocked, and I wouldn't know how to do this. So this is from the lab of Bruce McNaughton when he was in Arizona. Actually, was also my mentor. And so Bruce and Matt Wilson were the first using a technique that we call the tetrode to record many, many cells at the same time. And with this technique, for example, here, so they have 80 neurons recorded simultaneously. And now you see, how a population of cells is encoding space. Now, one thing you see is that many neurons have a completely blue firing map. So why is that? Because in this environment, they are completely silent, right? So only like 30% of the cells do something in a given environment. If we go in a different room, it will be another 30%. Okay? So a certain group of cells will be representing an environment. So you can, in this way, we can, for example, distinguish between things that happened in this room and things that happen in the next room. Okay? It allows us to separate, or when we use a fancy term, orthogonalize memories in different places. And so Matt and Bruce were also the first to decode position from the activity of these cells. And you can say, so they use some mathematical procedures, and they show with 140 cells, you can reconstruct rat position with an error of plus or minus 1 centimeter, if you use 1 second of activity. So that means that there is a lot of information. So in the rat hippocampus, there are probably about a couple of million cells. So if we can do this well with 140 cells, you can imagine that there is a lot of information. So now, why are these place cells so interesting? Well, they are interesting because they actually have to solve a very complicated problem. And the problem is that when we have to evaluate our position in space, we have to use a lot of different sources of information. We'll use vision, we'll use olfaction. If you are a rat, you probably use tact a lot. Rats have whiskers, which they move continuously, and they use to sense their environment. And that's probably their main sense of source of sensation. The other thing that is very interesting, important, is the use of self-motion, which means that they and we can update their idea of position based on how we move. So know that I'm here, so if I move 3 steps in this direction and then 1 step or 2 steps in this direction, I end close to the blackboard, for example, right? And that's why if you are in a very familiar environment, so like your bedroom, you are able to move around even in pitch darkness, So all of these things are combined together. And so what you have to do to study the contributions of these different components is do experiments where you change one source of input and leave everything else unchanged. So let's look at this experiment. So this comes from the lab of Bruno Pousset in France. And so here, we still look at firing maps. Okay, now the color code is different. So yellow is 0, blue is high activity. And here you see this cell that has this firing place field, as we say, where it likes to fire at the north end of the arena. And there are these 3 landmarks. So this was done in France. So guess what? These are 3 wine bottles. So you take the animal out of the arena, then back in. The cell is still there, still firing here. Take him out again, but now move the landmarks. Look what happens to the cell. Now it's here. So what does it mean? It means that it maintains a constant position, but relative to the bottles. And then what do you do? Well, you say, okay, now, oh my God, you broke this cell. So, well, we take the animal out, put the landmarks back to where they were. Place field returns here. And this is another example with a different landmark configuration, but same thing. Now, because now the place field is too far from the bottles for any olfactory or tactile cue to be involved, so the authors concluded that this would be about vision. Now you can say, okay, well, vision is the main driver of place fields. If you talk to John O'Keefe nowadays, that it was still what he would say. But here's another experiment. Empty arena, nice place field from the lab of Bob Mueller in New York. So what they did here was they just decomposed the activity based on head direction. So that means, so this is the activity when the animal is looking north. This is the activity when the animal is looking south, east, west, and so on. They all look pretty similar, even though the visual input, so if I look this way or I look this way, it's completely different. Okay, so it's not just vision. And another example. So this is the same cell recorded in the same environment while the animal is doing different things. So here, the cell was just recorded while the animal was randomly moving around in this arena because food was just sprinkled at random. In this situation, maybe half hour later, so the experimenter did a different thing. So they started putting food here, then here, then here, then here, and then here again. And then now in the same environment, now the animal does a fixed trajectory. And notice that the place field changes. So it was here, now it would be about here. Completely different place. Moreover, notice that now the field, this cell acquired an extra property that it depends on direction. So it would fire when the animal moves in the clockwise direction, but would be silent when the animal goes in the counterclockwise direction. So we learned a few things. So we learned that neurons, that the hippocampus can actually have multiple maps to encode the same environment depending on the context. So maybe I have a map to encode this room when I give a lecture here, which happens quite frequently, and a different hippocampal map to encode the same room when there is a party in this room, right? So, and that is a different organization of cells. So, let's say one more thing about the spatial system, and this is about self-motion or path integration. And this is an old classic experiment. So this is that with desert mice, species called gerbil. And so what they did here, Mittelstadt and Mittelstadt, they had a cage with a gerbil litter, a mom with pups. In darkness, there was a big circular platform. And then what they did was they took one of the pups and put it somewhere in the arena. And the mom would go searching for it in pitch darkness, finds it, and then what happens is that the mom would be able to just go in a straight line back to the cage. And if you were a little evil and move in the meantime, you would move the cage, the mom would still go back to the place where the cage was originally. Which means that essentially what she did was to track her own motion and track her position that way. Right? So, and I mean, experiments like that have been done with multiple species, including ants, that are actually pretty good at that. So it's actually something that's quite widespread. And so how does it work? So here is a little bit of vector algebra for you. So you have information about self-motion, and that's actually information about your direction of motion and velocity, or as if you want, is a speed vector. And so if you have the speed vector, so what you can do is just take the vector sum of it, and that tells you the total displacement, so how much you moved. So what you can do is then sum, or mathematically speaking, integrate all of these vectors. And then you can keep track of position. So that's why we call this path integration. So how do you do it? Well, we and others did neural networks models of that, and you can imagine that the neurons are organized in some sort of matrix, and then there is a bump. So a group of neurons that are nearby that are active. So if you look at the activity profile, it looks like there is a bump. That's why I call it activity bump. And with if you wire the network right in a way that is more or less biologically plausible, you can make this bump move with the self-motion. Now that was for some time the leading model of self-motion tracking. It has a small problem, and the problem is that you have a finite number of cells, and that, so what happens, so this cell will cover a certain surface. What happens if the animal gets out of that? Then the animal would be completely lost because he couldn't be able to track his position at all. So one way this could be working was if this had periodic boundary condition. That means if the bump gets out of this square from this side, it would come back from this side. So, which means that the bump would keep on moving. I would lose absolute position, but there is a way around it, which I will explain later. But that has the consequences that, so if the rat is walking a long distance, then it would get out, in and out of these boundaries multiple times, and the same cell would fire multiple times. So a cell might fire here, and then here, and then here, and so on, right? Well, this is not what we observe in the hippocampus. Hippocampal cells are non-periodic. Now, this is where the entorhinal cortex becomes interesting, because, hey, this structure has periodic place fields. So then the idea was, when these were discovered, that that could be the path integrator. And so we were among the first doing a mathematical model of that. So I will finish by going back to the idea of interaction between hippocampus and cortex. And I want to show some of my work from a few years ago, which is still similar to stuff we do recently. And so this has to do with a task where we made the rat decide whether to go left or right. And this was based on a rule, which means that sometimes you have to go always to the right, sometimes you have to go always to the left, so which means you have a spatially guided rule. But then there was also a little light that would come off on either this side or this side, and the other rule could be follow the light or go away from the light. And then whenever the animal would learn one rule, we would move to another rule. So the animal had to flexibly switch. Now, this part of flexibly switching is something that implicates the prefrontal cortex. So part of the brain, the part of the cortex which is to the front of the brain, which we use when we have to organize behavior, make judgment call. Interestingly, in humans, prefrontal cortex is the last to mature, and it's only completely mature around 22, 23 years of age. So which might explain something about youth behavior. So in any case, so what we did was to record from with this device, which at that time I was this was handmade. Learned how to make this stuff from a guy who was actually a watch repairman. So these days my students 3D print everything, so it's a little easier. And then you can look at how much these areas communicate by looking at, for example, at how much they oscillate in synchrony. So now if you look at the activity in each brain area, you see waves. So that's what you see in the electroencephalogram. So if you go to a neurologist, so that's what they will tell, and they have to test you, they will see these waves. They will put electrodes on your scalp, and they will see waves, and they can actually diagnose epilepsy and a lot of other conditions. So we do the same, but with inside the head. So we can get precisely to precise brain area. We can go to hippocampus, prefrontal cortex, and we look at how synchronous these waves are. Now, the synchrony actually changes during the task. And it turns out that it's maximal at the fork of the maze. So what's the fork? It's the decision point. So that's where the animals have to decide, shall I go left or shall I go right? And actually, they really kind of decide there, because if you look at the behavior, especially at the beginning when you're training them, so they will do this. They arrive at the fork, and then they start looking left or right and deciding, right? So we saw that kind of, that these two areas actually get engaged with each other while the animal was making the decision. And that turned out to be also learning dependent. So we'd only see this after the rule was learned, and much less when the animal was just performing at random and trying to figure out what the rule is. Of course, we never tell the animal what the rule is. They have to find out by seeing which time they get rewarded and which times they don't. The other thing we did was then looking at memory replay. And actually, we found it. So this is what looks like. So it's a little complex slide. But basically, this trace tells you the intensity of this replay signal, which we find with a mathematical procedure. And you see that there are a lot of peaks. And this is the sleep after this experience. This is the sleep before, which we use as a control. You see there is not so much there. And we have to do that because we have to make sure that these are not patterns that are there just all the time. So they would have no link with the experience. So the colorful background, so besides being nice, it's a representation of the EEG of the animal. And these yellow stripes correspond to periods when the animal was sleeping. So you can see that the peaks are actually exactly in the periods of sleep and not so much otherwise. And the main result of this study was that, so this is the cortex, but bursts of activity in the hippocampus that we call the sharp wave are actually related with increased replay in the prefrontal cortex. And this is interesting because these bursts of activity that we study a lot, these sharp waves, are where the hippocampus itself is replaying. And therefore, so what we established there was actually for the first time at that time, a connection between the hippocampal and the cortical replay. And so the idea was to go towards a study of this phenomena in the entire brain, so in the global brain, which is what we are currently trying to do in the lab. And so with that, I would finish. And so if there are questions, I'd be happy to. Yes?
What about the amygdala?
What about the amygdala? Haha. Okay, so the amygdala is, I worked a little bit on that in a collaboration with a colleague. So the amygdala is considered to be very important for emotional memory, so that it's where memory about emotional occurrences would be stored.
That's also that you say at the beginning, episodic memory.
Yes.
Yeah, it's more emotional.
Yes. And indeed, so the amygdala is very tightly connected to the hippocampus. And it's very tightly connected with a number of the structures that are connected with the hippocampus. So we talk about the limbic system, so which is a set of structures that have a connection to emotion. The other interesting thing is that all these structures have a connection with the internal state of the organism. So they get information about current stress levels, heartbeat, and so on and so they hunger, and so all this information that comes from a place way deep in the brain called the hypothalamus. So the combination of this information about the body and the emotion is a very powerful way to steer memory itself.
Well, also we need to talk about this because episodic memory goes back to 3 or 4 years old. Then you are able to we call it.
Yeah.
Yeah. But the emotional memories
Yeah.
are going further.
Yes.
Was to birth or maybe even before birth. Yeah.
Yes. Actually.
I want to say, I was thought maybe you would also talk about the EMDR.
That is not really my field. There is a colleague here in the faculty who's actually looking at the memory system in rat pups. And so they are actually going to the exactly this period of infantile amnesia. So she's doing very challenging experiments to record neurons in those pups. And for the time being, she is looking at the hippocampus, but maybe she will get in the, yeah. But so it is a very interesting thing. And so one thing is about these sharp waves that we are very interested in. So those seems to mature a little bit later in the rat's life, and that's might be where this autobiographic memory might start. So, but, yeah. Yeah.
Can you elaborate a little bit on reliability of memory?
You mean?
I'm referring to getting something out of memory is restoring it at the same time and thereby changing it slightly. That's what I've read somewhere.
Yeah, well, two things. So, when you said reliability, I thought in forensic terms. So, no, because that is actually, it's also a field of study. Like, if you have witness in the trial, how reliable those memories are, right? So, and,
We're going to the rats.
yes.
If you repeat experiments several days later.
Yes. Okay, so the best, now the neural memories for like the Morris water maze, for example, so those can be maintained for months. If you look at the neural code, so the best data are actually from mice. And actually, you see that in 2-3 weeks, the spatial code that encodes a certain environment changes completely. So that was done not with the tetrode, so this technique I showed you, because with that it's very difficult to see the same cells across. They was done with the calcium imaging, so that thing I showed in the previous talk. And with imaging, you can see the cells, and then you can more confidently say, okay, it's the same cell 12 days. And that in that situation, in mice, it's like 2-3 weeks, which is interesting because, indeed, mice can maintain spatial memories for longer. So it's a little bit like you store information on the cloud, and then maybe one day Amazon puts it in a server in Ireland, and maybe in 2 months it's the same information, but it's in a server in Canada. And, so there is a way that the brain can do it in a transparent manner, apparently. Yeah.
If you look at navigation skills, some of my friends are very good at it.
Yeah.
Others are very bad at it.
Yeah.
I think the same might apply for mice and rats. Or is that?
Well, yes. There is a distribution of navigational skills across individuals, and there is also a link with the precision of displaced cells. So the one thing where this has been studied best is with aging. So you can study 3-month-old rats, or 1-year-old, or 2 years old, which is about, it's like an 8 years old human, and then you see the decay in both spatial memory and place cells, activity, resolution, let's say. We also work with transgenic models, so where you can, with genetic techniques, interfere with the function of the hippocampus in one or the other way. And that's another way to study this thing. So one thing you have to say, so the individual variability is less because we use rat strains that are very inbred, So, yeah. What's another? Okay.
To what extent can results of your research be used in the area of artificial intelligence, where you are also constructing neural networks? Usually these are much more simple ones, but maybe having their different architectures would lead to better results.
Yeah. So many ideas from in artificial intelligence are brain inspired. So when we talk about deep learning, so these ideas that there are multiple layers of neurons, that is something that is inspired to the cortical architecture. For example, this idea of replay, now it's something that is being included in algorithm by Google and others. So there is, so Google in particular has a division in London called DeepMind that they are essentially neuroscientists and they're trying to apply a lot of this knowledge. So, yeah.
Memory stored in genes. In which part of the brain those genes are located?
Well, genes are the same in all cells. So what changes is the pattern of expression, so which genes are expressed in each cell type. So your skin cells have the same gene as your neurons. It's just that some of them are activated in your skin cells, and some other ones are activated in the brain. So how? And each brain area has its own gene expression patterns, and so these gene-encoded memories can be very specific to one brain area. Now, studies of these things are typically done with fruit flies, Drosophila, because there you can very easily alter the genome and do it on many individuals. So that's the kind of study, but you can get, so you can imagine that, for example, you can alter a reflex in the fruit flies by altering a gene. And these kind of things have been done. It's time? Okay. Well, then we finish here. Well, thanks a lot.